We are looking at the gastric mucosa. This is what the tumor looks like at low power, a big solid mass. This is what the tumor cells look like at high power. Some are elongate, some are round. We need to identify what type of tumor that is. And in the laboratory, we do a variety of ancillary studies. You've heard the term immunohistochemistry. And what immunohistochemistry is, we take advantage of the fact that we can make a molecule that recognizes a certain fingerprint. And that fingerprint is the protein c-KIT. And so when we subject that tissue to the tumor, it binds to the tumor and we put a visual tag at the end of it. And when it's brown, that's where the visual tag is. That means the molecule that it's staining is c-KIT. And putting all the clinical findings together, what the tumor looks like under the microscope, what type of proteins it is producing, I can make a definitive diagnosis of a gastrointestinal stromal tumor. I look in the medical record, or not. Also, obviously, I received the specimen. I know where the tumor arose. I know how big the tumor is. I spend maybe 20 minutes looking at it, trying to do a very careful assessment of how often the tumor cells are proliferating, and that we translate into a mitotic count. And putting all those features together, I can provide a risk assessment of how that tumor is likely going to behave, so my clinical colleagues can devise the appropriate treatment team. But is that enough? No, and obviously that's not enough. And what I'm going to be talking about, again, is the role of mutational analysis and NGS in gastrointestinal stromal tumor. Mutations in gastrointestinal stromal tumor and understanding the molecular biology is foundational. Foundational in a sense, just as a genetic disease. Understanding the genetics provides information. Why did I get a gastrointestinal stromal tumor? Why was it the ileum rather than the stomach? It is the genetics that determines how big it's going to get, how fast it's going to grow, whether it's going to get into the lymphatic system, how fast it's going to spread, and if it does spread, how fast those deposits are going to grow. So it is the mutation, the genetics, that are extremely powerful and cause the problem, cause the disease. So I like to start out very basic concepts. So I want to provide you with definitions. So what is a mutation? So a mutation is defined as any change in the DNA. And I will get into that a little further. You hear the terminology NGS, next generation sequencing. What the heck is that? Well, what next generation sequencing is, it is a type of technology that's extremely sophisticated, extremely powerful, that assesses every little molecule in the DNA to determine whether or not there's a mutation. And this is the type of equipment that is or performs the next generation sequencing. So when we talk about DNA, remember, we all have DNA, and DNA is like the brain for our cells. It is the blueprint for everything the cell does. And the cell always follows the blueprint. And the DNA is composed of molecules. And look at some of these numbers. In just one cell, and I may have, again, 10 trillion cells in my body. one cell, excuse me, I think, there we go. There are 3 million molecules that make up the DNA. So when you talk about next generation sequencing, that machine is looking at 3 billion molecules in the DNA to see if one is just wrong, or maybe 10 are wrong, or 1,000 are wrong. So extreme large numbers of cells. And remember, these molecules that make up the DNA in the human are aggregated into structures which we call chromosomes. And in humans, we have 46 chromosomes, but each chromosome has hundreds of millions of these base pairs. And the next generation sequencing is looking for an abnormal base pair. Think about gastrointestinal stromal tumor. A piece of the tumor, literally this big, has over 1 billion cells. Our cells try to do the best they can to keep the DNA intact. Because once the DNA changes, mutated, it's potentially a really big problem. That's how cancer develops. There's a mutation in the DNA that infects the entire cell, changing the biology of the body. So I want to put it in further perspective. 3 billion base pairs in our DNA. We are looking at a diagram of a cell. We can't see just one cell with our naked eye. It's too small. But there are 3 billion base pairs. This is the diagram of a cell. This is where our DNA is. This yellow covering, these are proteins that surround the DNA and interact with it. And why am I telling you this? Because for those patients who have SDH and they're talking about methylation and epigenetics, it's problems with some of these proteins that can affect the DNA and cause the same type of abnormality in a practical sense as a mutation in the DNA. The cell has to live. It has to produce energy. These structures in the cell are known as mitochondria. That's where energy is made. Why do I talk about mitochondria? Because we've identified mutations in proteins in the mitochondria that results also in GIST, and those are the SDH deficient GIST. And lastly, you can see here our diagram of c-KIT for everybody to understand where it is. It is on the surface of the cell. Imagine I'm a cell. My arm is like a c-KIT molecule waiting to bind something to make that receptor do what it's supposed to do. Now I said a mutation is a change in DNA. There are different ways to change the DNA. We can lose maybe 7 base pairs in a certain segment. That's known as a deletion. That's what's happening in many of the mutations in the gene that codes for c-KIT. A group of the base pairs are lost. It changes my arm and maybe makes it look more like a leg, and so it acts differently. We can have a kind of change where we don't lose DNA, but there's a change in the base pair, one replacing another. An example would be, here's my hand, you replace my thumb with a toe. It changes everything. And that's known as a substitution. You may hear about insertions, new base pairs that were never there being put into the DNA so it changes the structure. And then lastly, you hear about fusions or translocations. What that means is a piece of DNA from over here was moved and inserted into the DNA here. And the piece of DNA that was here went to this portion of the chromosome. So there is a change in location of aspects of the DNA, and that changes the function and activity of the cells. So it is this type of information which is basic to understanding the biology of your tumors. Imagine this is maybe 30, 40,000 dominoes. This is small compared to the number of base pairs in our DNA. We have a change in one, just one, can throw everything awry and change how that cell functions, how often it divides, how long it lives, whether it can get into the vascular system or not. This is what mutations can do in sort of a fantasy way. But this is also what mutations can do. It produces a cyst in the ileum that potentially could be fatal if it's not successfully treated. Another way to look at a mutation, it's like a conductor. The conductor is telling the instruments what to play and when to play. You have a mutation in the conductor, and again, in the analogy to the cell, each one of these molecules is one of the musicians working together to allow the cell to live. If we get rid of the conductor or change the conductor, if we change one of these molecules in many of these very important pathways, again, it changes the biology of the cell. And all of that is in play in GIST. And in fact, although it is very complicated, as we understand more and more about the genetics, it creates wonderful opportunities to treat the diseases. Because if we can identify the genetic abnormality, it gives us an opportunity to discover and implement a cure that works against that mutation. So these are the names of the mutations in the vast majority of gastrointestinal stromal tumors. And as you all know, the almost 75–80% of all mutations affect the gene that makes the protein c-KIT. And again, c-KIT is a receptor molecule sitting on the surface of the cell. We then have our next big group of the SDH deficient GIST, different mechanism of causing the cell to be a cancer cell. Mutations in BRAF, KRAF, neurofibromin, and then we have these fusion genes which I talked about, many of them associated with translocations. These account for a very small percentage of GIST, but the reason why they're important to recognize is that some of them may be effective with the current drugs that we're using. And understanding how these result in the cancer GIST can allow for more new discoveries that potentially therapies that can affect GIST regardless of the type of mutation. So I just want to look at a couple mutations in a slightly greater detail because they affect many of the patients who have GIST. As I said, c-KIT, the molecule, this is what it looks like if you were to try to draw a model of it. The genes, the millions of base pairs that code the information for c-KIT are located in chromosome 4. c-KIT is a very important molecule in normal cells, certain types of normal cells. And its role, you can see, it's involved in cell survival, proliferation, adhesion. Adhesion means being able to stick to where it should be, that cell, and as well as that cell appropriately maturing and becoming a functional actual cell. These are diagrams of c-KIT. This is the cell surface. This is the normal molecules of c-KIT. Normally, with regards to c-KIT. I have them on my body, on my cell. Those receptors are waiting to bind a substance so the two c-KIT molecules come together and activate the molecules in the cell that allow it to live and do what it's supposed to do. What happens when we have a mutation in c-KIT, when we lose certain base pairs, those molecules start acting on their own. They're activating the cell in an abnormal way, causing all of these pathways to be active when they shouldn't. And we can get mutations in different portions of the DNA that code for the molecule c-KIT. And we know that the different mutations can be very important with regards to how aggressive the tumor is going to be, how it is going to be responsive to imatinib and other potential drugs. Understanding these mutations, how they affect the biology of the tumors, allowed us to identify certain types of GIST are more aggressive or less aggressive compared to others. You know that, again, the vast majority of mutations in GIST are affecting the genes for c-KIT and platelet-derived growth factor receptor, that approximately 12% are considered wild, meaning they don't have a mutation in c-KIT or platelet-derived growth factor, but a mutation in one of these other genes that also can produce a tumor that otherwise is like GIST. And the one that I just want to spend a minute on is the SDH deficient GIST. I want to go back to this diagram again. The SDH, the succinate dehydrogenase enzyme, is located here within the membrane of the mitochondria. And it's very important in the producing energy within the cell. And when there is a mutation that affects this enzyme, it prevents the normal conversion of one type of molecule known as succinate into another type of molecule. And if that conversion is blocked, the number of molecules of succinate increase in concentration within the cell, and that affects the DNA. And it results in alteration of the DNA, which many of you may have heard metallization, meaning new molecules are attached to the DNA that interfere with its normal function. And there are a variety of different aspects that are related to the SDH deficient GIST. And importantly, as you see here, they're not responsive to tyrosine kinase inhibitors, and people are actively trying to identify what kind of therapies conceivably could be used to treat these patients. You've heard of the epimutant SDH. What that means is the DNA is okay, and we're looking at the DNA here, a graphic of a cell, but the proteins that envelop the DNA, there's a problem with them. And it's the problem within those proteins which control what portions of the DNA are going to be expressed that interferes with function, resulting in the generation of a gastrointestinal stromal tumor. In addition to identifying the mutations that are felt to be responsible for the formation and biology of the GIST, there are a variety of mutations that affect other chromosomes and other genes that can develop over time. And that's what normally happens in cancer. Cancer is, the cells are not static. The DNA just doesn't have one mutation and it stops there. Over a period of time, new mutations develop. And those new mutations can be very problematic because they can make the tumor more aggressive. they can activate the cell pathways that prevent a drug from being active. So maybe imatinib worked for the first 5 years, but now it's not working. Why isn't it working? Well, it's very possible additional mutations occurred that prevent it to function the way it should. And so genetic analysis can provide that information to provide the clinicians an explanation potentially why certain drugs are not working, and if these new mutations have developed, is there an additional type of therapy that can address the new genetic abnormalities. So, with regards to genetic testing, it's my opinion that all tumors, all GISTs that are intermediate or high risk should undergo mutational analysis. That the genetic information that we learn obviously provides very important information related to the potential prognosis associated with that tumor, whether that tumor is going to be responsive to the drugs that we currently have, and that type of information again provides valuable data of how the DNA is changing over time. So maybe mutational analysis is not just done at the time of diagnosis, but also later on in the course of the disease to identify the new genetic abnormalities that have occurred. And lastly, this type of genetic information for the small number of patients who have a syndrome, that can be very valuable because of family members that would potentially need genetic counseling because they may be at risk for the disease as well.